Perfect. Thanks, Rich, for the introduction. And I think this is going to be a really wonderful session. A lot of work has been just putting this together, and I think we're going to see some really cutting-edge research. Again, the talks for total can be up to 15 minutes. You know, if we have time for a couple of questions after each one, we will take it. If not, there is a 10-minute reserved panel discussion, but we will stay on time throughout this morning. The first speaker is Dr. Sergei Doulatov from the University of Washington, who will be talking about iPSC and CD34 positive cell models of SF3B1 mutant MDS.
All right. Okay, great. So thank you to the organizers for having me come and speak here today. It's my pleasure to tell you about our new models of SF3B1 mutant MDS. So I don't think this audience needs a reminder about this, but my lab is very interested in modeling this progression from normal hematopoiesis, from normal hematopoietic stem cells, through kind of these premalignant stages like clonal hematopoiesis and clonal cytopenias, to myelodysplastic syndromes and ultimately acute myeloid leukemias. And in particular, we've been using two different approaches that I'll talk about today to develop these models. One is on the left-hand side, using induced pluripotent stem cell reprogramming, so taking cells from the marrow of these patients, reprogramming them using Yamanaka transcription factors, and then establishing pluripotent stem cell lines as a tool for disease modeling. And then the second approach that I'll discuss today, kind of our newer work, is by CRISPR editing primary CD34 positive cells to establish these models. So talking about the IPS models a little bit, one of the things that we found early on when we started this work is that when you reprogram a complex mixture, heterogeneous mixture of clones from the marrow of these patients, you don't just get a single clone. You get clones corresponding to all the different sort of clonal intermediate stages in this disease. And so, for example, taking an MDS patient with 4 different driver mutations that are listed out there, we can get IPS clones corresponding to all these different genotypes. For example, single mutant, double mutant, triple mutant, and so on. And you can see that these clones kind of nicely capture this clonal progression that we see in this disease. What was really striking to us is that when you compare that frequency of these clones to what we get directly out of this marrow, for example, just using colony-forming units, iPSCs are actually very good at capturing some of these earlier clones that are quite rare in the marrow. And so there's something about IPS reprogramming that was very informative and instructive for capturing these early clones. And of course, by direct deduction, we can then establish the order of driver mutations by just examining the genotype of these IPS lines. So this is a nice model in the sense that we get all of these genotypes that occur during disease progression. And now we've established quite a repository of these different IPS lines from many different patients, sort of capturing all of these intermediate clones. And these are, we're trying to make these available to the community, and I'll explain a little bit about kind of how we can make these easily available to all of you. So let me talk just very briefly about SF3B1 because this hasn't been mentioned yet, I think, at this meeting. So splicing factor mutations are very common in MDS. It's one of the most common mutations, including as well as other splicing factor mutations. And in myelodysplastic syndromes, they're typically associated with favorable prognosis and the presence of a ring sideroblast in ineffective erythropoiesis. And there's other co-occurring mutations that can confer poor prognosis. So we've started modeling this early on because as you know, many of the models that have been previously developed, for example, the knock-in mouse models or the human cell line models, don't recapitulate some of the features that we were looking for in this disease. For example, the presence of ring sideroblasts, which is kind of a hallmark feature of this disease that hadn't been previously captured in these models, and that's what motivated us to start developing these models. So the IPS models that I'll just talk about briefly because this has been published, but we see very nice ring sideroblast formation, and this is kind of what launched us into this. And the CRISPR editing model, we hope, can capture some of the more stem cell biology in this disorder. So how do we work with these lines? They're technically kind of challenging to work with. How can we make these available to the community? Well, what we've been doing is establishing cell lines using these 5 transcription factors from these IPS models. And these basically propagate as CD34 positive cells that we can keep in culture with doxycycline. And they can still differentiate into all the different lineages and as well have some transplantation potential that I'll just show in the next slide here. But first, I just wanted to show this nice picture of ring sideroblasts that we get from SF3B1 mutant IPS cells when we direct them to differentiate into the erythroid lineage. And these are stained using pearl stain. And from this, we've been able to dissect out the pathophysiology of this, which has to do with aberrant splicing of these 2 different genes, TMEM14C and ABCB7, that are both involved in iron handling and metabolism in the mitochondria. And having this model had been instrumental in being able to kind of dissect out this particular phenotype. And so this is kind of what I mentioned before, that we can, these cells are not just useful for in vitro differentiation. We've been able to take these and transplant them into immune deficient mice, and then they generate quite a robust engraftment in vivo. And so, for example, here taking 2 different lines from a single patient, one that has a RUNX precursor mutation and the second RUNX SF3B1, we can get quite reasonable levels of engraftment. And the cells coming out of the marrow have this immature myeloblast phenotype. So we hope that then actually I will show how this model can be used primarily for drug studies because obviously it's very difficult to conduct these experiments with primary MDS patient cells. So what motivated us, however, more recently to get into the to gene editing of primary CD34 positive cells is that for a long time we've been trying to get these iPS-derived cells to specify into real bona fide hematopoietic stem cells, and that's still been a very, very difficult challenge. So to try to understand what these SF3B1 mutations are doing in the context of normal hematopoietic stem cells, how do they transform hematopoietic stem cells? Because we know that they occur very early in clonal hematopoiesis, early in the pathogenesis of MDS. So we've developed this gene editing system which uses the AAV6 donors. So these are, of course, gain-of-function mutations, so we can't just knock them out using standard CRISPR technologies. So we need a donor template to introduce the mutation. And so we've been using the Majetti-Porteous developed AAV6 donor with the SF3B1 mutation included in the donor. And we've made some changes to this construct that allows us to basically integrate this, and I'll show this in the next slide, into the endogenous SF3B1 locus. And so the questions that we're hoping to ask that I'll just go through quite quickly is sort of what are the targets of SF3B1 in stem cells, and does that expose any kind of therapeutic opportunities for us to be able to target them? So yeah, so this construct has a traceable allele. It has a BFP tag that goes into the intron of the SF3B1 gene, and it has the K700E recurrent mutation in the right homology arm. And so that enables us to edit directly in the endogenous locus. And you can see the editing is heterozygous, and it's fairly efficient with about 20% editing in the CD34 positive cells. We first asked how do these cells behave in culture. So you can see that looking at the stem cell compartment is fairly well maintained. But when we put these cells into erythroid differentiation, we see an effect of erythroid differentiation, just like with the IPS lines that I've shown you. And we do get some ring sideroblasts, although there's something about IPS cells that's particularly good at making them. And so really what we wanted to know, it's what's downstream, what is SF3B1 targeting? And this is just taking a very kind of broad overview of this. A lot of the genes that are aberrantly spliced in the presence of these SF3B1 mutations in stem cells have to do with cell cycle, mitosis, and genome maintenance. So this may not be surprising to some people, but it was really surprising to us to see this really strong signal of these genes being aberrantly spliced and also downregulated at the transcriptional level. And we see the same thing with single-cell RNA sequencing. It's a little bit more informative here because we can trace what happens to these cells, to their differentiation. They kind of start out here where you see HSC there. And normal cells, sorry, you can't see with my pointer, normal cells kind of migrate towards this kind of masked lineage on the left-hand side, but these SF3B1 mutant cells kind of stay in the stem cell compartment and predominantly try to differentiate down the erythroid lineage, and this is kind of where they fail early on. So that was quite interesting to us. But molecularly, we kind of see the same signal with these cell cycle, mitosis, and genome maintenance genes being downregulated specifically in the hematopoietic stem and progenitor cells. And so that was an interesting signal, and it does expose a therapeutic vulnerability. And so I won't belabor on how we came upon this, but what that does is it creates this incredible vulnerability of these cells to DNA damaging or agents that perturb DNA damage and mitosis. And one of these is CHEK1 kinase inhibitors. And so we found that these cells are incredibly sensitive to CHEK1 kinase inhibitors like this inhibitor prexacertib that's been in clinical trials. And I'm including the splicing inhibitor pledenolide B there as essentially just a reference because these have actually made it into humans, into patients. And you can see that SF3B1 mutant cells with many different genotypes are incredibly sensitive to these CHEK1 inhibitors. We've also looked, what the system also allows us to do is to look specifically at how the stem cells are responding to these inhibitors. Because if we kill progenitor cells, that's not terribly helpful. What we need to do is kill the disease-initiating cells. And so we can look at that by using our system. And when we look at bulk CD34 positive cells, they actually respond fairly well to both SF3B inhibitors, so the pledenolide, and the CHEK inhibitor. But if you look at stem cells, the phenotypic stem cells that are marked in culture, they're actually, when treated with the splicing inhibitor, this inhibitor is actually quite toxic to normal stem cells. So it's very difficult to fine-tune and to find the right dose that would be effective. And I think that tells us a little bit about using these compounds in patients. Whereas when you treat them with CHEK inhibitor, with prexacertib, you can see that normal stem cells really don't care about it, and it's selectively acting on these SF3B1 mutant cells. And then going back to the IPS model, as I've alluded to, it's very hard to find, to be able to do these in vivo experiments. We've turned back to the IPS model where we've taken those SF3B1 RUNX mutant cells that I introduced to you, and I already told you that they could be engrafted in xenograft mice, and we transplanted them and treated these mice with prexacertib. And so what you can see is that vehicle-treated cells have low but very appreciable levels of engraftment, and this engraftment completely disappears when treated with prexacertib using established preclinical regimen for this compound. And this is just some immunophenotype from these mice. So, this model has been incredibly useful for these kind of in vivo drug experiments to establish proof of principle. So let me just summarize. I know I went through this quite quickly, but we think that the IPS is a robust platform for preclinical disease modeling and allows us to basically test things in individual clones, genetic subclones derived from either CHIP or MDS. And these cells can be transplanted into NSG mice and could be a useful tool for these drug studies. We've also established precise gene editing of human CD34 positive cells. So these are cord blood or peripheral blood derived CD34 positive cells, and shown that they could be useful for disease modeling in vitro and in vivo. And using this pipeline, have identified CHEK1 inhibitors as a promising therapeutic option that has been well tolerated so far in trials. So we think it could be promising for maybe high-risk patients with these mutations. So with that, I'll end. I'd like to acknowledge people in my group that have done the work and our collaborators, Rob Bradley at Fred Hutch and Luca Molcavati in Pavia and our funding sources. So I'll take questions afterwards.
Perfectly timed. Any burning question? We maybe have time for just one question from the audience. We got right in the middle, and then we'll move on. Down one row in the middle.
Marcin.
No, beautiful work. One question about the engraftment capacity of the iPSC-derived hematopoietic progenitors. So because that's obviously an issue in the field, right? So what percentage of engraftment do you achieve with wild-type cells, and is it affected by the mutants?
Yeah, it's a great question, Marcin. Thank you. It's, as I alluded to, it varies from line to line. So, for example, with these two lines that we've studied, that are the RUNX and from this patient that has RUNX and RUNX SF3B1, the RUNX line engrafts almost 80–90%. It's an incredibly robust engraftment that's long-term. I also didn't explain that. But the SF3B1 clone engrafts at a lower level. And so, we don't know if that's the effect of the SF3B1 mutation or it's kind of a line-to-line variability and heterogeneity. But I think that kind of shows the range of these. And, we need to do more work. We've obviously proposed to and have been distributing these lines to investigators. So I think we need to do a lot more to kind of establish how they engraft and, yeah.
So once this line is established, do you have to retransplant, or can you then, let's say, if we put it into mouse, we can then serially transplant in perpetuity?
Yeah, that's another great. We haven't tried serial transplanting. I would imagine that if it's robustly engrafted in the primary receipt, you can retransplant those. So I think these will be useful for people in the community.
If you are being asked, well, can you publish these cell lines, supply them to us, you would give to the collaborator, the IPSS, and tell him how to do it, or you would give the differentiated cells?
No, we would give the actual, we've been distributing the actual lines. And that's kind of what I was saying, is that the IPS themselves are kind of tricky to technically work with. Not a lot of people have that capacity, but these cell lines are pretty easy to set up and establish. And, they're a little bit harder than K562s, but not that much.
Okay, thank you. Next presentation is by Dr. Babal. He is going to talk about the most common mutation in myeloid neoplasia, namely TET2, which he has made a subject of his life for the last 5 years, 10 years?
10 years. Okay, thank you, Jarek, and thank for the organizer for giving us this opportunity to share some of our recent work. Some of them we have published and some of them we haven't. There's some of the fresh data which is under some of the manuscript. Now we know this audience is not does not need an introduction to the idea of somatic mutation which drives malignant evolution, and majority of this MDS and their progression into AML is driven by some kind of somatic mutations. While these somatic mutations drive malignant evolution, they are also critical. They provide some kind of idea how to target them specifically and how we can differentiate between a normal self as well as the malignant mutated clone. Now, one of the key parts which we have observed, the majority of the mutation involved, somatic mutations involved in MDS and AML, they converge to the epigenetic dysregulation, like either this is a methylating enzyme or demethylating enzyme like TET1, 2, and 3. And this is one of the work we did with Jarek, it's a few years old, where we have seen that as age in cancer risk increases by age, it's not a new thing, but we did a whole number of search in the literature, and we did some analysis of looking at the correlation between methylation and cancer incidence. It's striking that as we age, the methylation increases because of the fitness of some of these demethylating enzyme. And we did over 1,000 MDS patients, and we saw that if you are 90+ year old, the chances that you will be a TET2 mutant is almost 90 to 100%. That means that there is some method to this madness of clonal evolution of loss of function in TET2 and the MDS. Now, what TET does, the TET is, for us, TET is the guardian of transcriptional universe. Because there are 3 TET enzymes, TET1, TET2, and TET3, and these enzymes regulates efficient transcription by demethylating the promoter enhancer, which are methylated under as a default conditions, and the demethylation is critical for recruiting the transcription factor, and hence they lead to their transcription efficiency, and that determines or that mounts accurate transcription to create homeostasis. And any defect or any perturbation in this either will create a skewed differentiation and in some cases malignant evolution. Now, as we know that I said that the TET proteins are TET1, 2, and 3. One of the most common feature of these 3 proteins are they have a conserved, almost identical, 75% identical catalytic domain, which is in every case it lies on the C terminus. But the N terminus, we did analysis for the N terminus domain, which has not well studied in either 3 TETs, but TET1 and TET3 has a well-defined DNA binding domain, and TET2 does not have a well-defined DNA binding domain. Thus, we have previously shown that the TET1 and TET3 are processive TET dioxygenases, while TET2 being more promiscuous. When I say TET2 is more promiscuous, that means it is more like a hit and run, and it creates more 5-MC to HMC compared to while the TET1 and TET3 leads to a processive oxidation of methyl cytosine and leads to the complete oxidation till CAC. Now, there are number of theories proposed how and why TET2 mutant clone evolved into malignancies. Overall, there are 2 major mechanistic links which has been proposed earlier before we started working on this in this area. That one was these, because if you lose TET2, there's increase in methyl cytosine in the genome, and that leads to more C to T transversion, and hence you have a bigger chance of second hit, and that second hit theory leads to malignant evolution in terms of MDS and AML. And another thing is that since TET enzymes are critical for forward differentiation into the mature blood cells. If you lose TET2, there is a myeloid skewing, and at the same time there's a differentiation block, and that differentiation block leads to the replication penalty, and hence there's immature blood cells, and that causes MDS and AML. Now there is these 2 theories has been partly in a number of different papers. It has been shown that in part it may be true. To test this idea, what we did, we took TET2 wild type mutant or the heterozygous mice, which we have, and we treated them with the carcinogen like ENU, and then we found that as expected, the TET2 double mutant, almost all TET2 mice developed AML, MDS, MPN kind syndrome, and some of them also had lymphoid proliferative neoplasia, and we confirmed it via a number of different assays and looking at the c-KIT positivity, WBC, and their splenomegaly. Now, we further went there and we isolated the LSK cells from these mice who developed leukemia in TET2 wild type and TET2 mutant. And we did whole genome sequencing coupled with the bisulfite sequencing, and this paper was published a few years back with the Ming Zhang from, now he's at UT Southwestern. The most interesting part there we found was the majority of the mutations in TET2 mutant clones were around the 5-HMC locus. Counter to the previously prevailing idea of C2T transversion, in the mouse model, this was shown that the majority of the mutations were this last red bar, which is around the 5-HMC locus. And that was a little bit surprising to answer why it is happening. So what we did in the lab, we developed 3 independent model system to look at the TET2 interactome. What are the TET2 proteins TET2 particularly interacts with? And we pulled down, the most interestingly, what we found that one of the, in every single set system, I will show you more data in next few slides, the MSH6 was the constant binding partner of TET2, and MSH6 is a protein which is a critical component of mismatch repair complex, which recruits MSH2 and MSH6 and then does G2T, corrects the G2T mismatch. And continuing with that, we confirm it in a number of different type of cellular model, either murine ESC, or we look at the 293 system, or the HeLa cells, or we look at the K562 cells level. And we confirm that this TET2 interacts directly with the MSH6. And then we did series of serial deletion in the TET2 construct, and we found, interestingly, it's not the catalytic domain of TET2, it was the N-terminus domain, which is in the middle of the TET2 N-terminus, which is around 400 amino acid to 1,000 amino acid. That segment is critical for binding to the MSH6, and this we did confirm it in a number of different assays, suggesting that, indicating that the TET2 non-catalytic function may be important for these for this mismatch repair complex recruitment around 5-HMC locus. And then this is the whole genome, whole exome sequencing data, and we analyze them. And interestingly, the most interesting part which I want to highlight, when though the sample size are small, but what we found is that when there is a frame shift deletion or double TET2 deletion or highly pathogenic deletion of TET2, there was a higher SNV per megabase compared to when we have a missense mutations. And the missense mutation which is expressed and which is that in our mind and our understanding is that maintains the genomic fidelity by recruiting mismatch repair complex around 5-HMC, even in absence of TET2 activity. Now, thus we have this model that what happens is that TET2 not only converts 5-MC to 5-HMC, it also involved in the repair, in basic sin repair part by recruiting mismatch repair complex. Now, to conclude that this is said that all TET2 mutations are not same, and this we are still working on how in clinical setup how this different type of TET2 mutational configuration results which clonal chip clone will progress or not progress, and we are looking at that aspect currently. Now, one thing is very interesting, that TET deficiency is one of the major, most frequent event in MDS and AML. So when I started my lab in the department which Yarek heads, idea was that can TET deficiency be targeted? And I come from a structural biology background. I do crystallography and structure-guided drug design. So one of the key idea was that time when we came, Yarek wanted us to develop some of the turbo substrate, or he was calling it something which can restore what is lost. And it was very intriguing and very interesting idea that time, and then we started working on this concept that can we design a small molecule, because since it requires alpha-ketoglutarate and iron 2+, can we create a turbo substrate activator of TET? And I will show you some of the data what happens next, and we spent a lot of time. And the second idea was, which we came simultaneously, which we have been pursuing despite knowing that it is very challenging, is that the TET, there are 3 TET enzymes, TET1, TET2, and TET3. Whether the TET2 loss creates synthetic lethality for the further TET inhibition in TET2 mutant clone, while giving a clonal advantage, or giving advantage, not a clonal, but giving advantage to the TET2 proficient normal hematopoietic stem cells. Now, first aspect I'm going to show that when we were working on this, and we found that whether we can restore TET activity, and we can find this restore the terminate the malignant program. And with the Ming Jiangzhou, we collaborated, and because for mouse, ascorbic acid is not a vitamin, because it has an enzyme, gulo enzyme, which makes vitamin C. And then we crossed the gulo mouse with TET2 to create a vitamin C deficient model. But even supplementing supraphysiological dose in the context of mouse, we don't see much difference. And most importantly, when we look at the human primary cells, we were hardly able to see any of this difference. Now, when we further went back and then we published why the vitamin C does not work, because vitamin C is a known redox which converts Fe2 to Fe3+, but the loss of function mutation in the catalytic domain happens because of the acetylation of lysine, and that does not, then if in that case, if we overload with vitamin C, it does not make any difference. And these lysines were across the board was conserved in different TET protein. And on top of that, we developed a very potent activator of TET, though it restores TET activity in malignant cells more than 30-fold, but it does not terminate the malignant program. And then we concluded that restoration of TET activity is not sufficient to terminate the malignant program when the AML has been already produced. So then we tested the second hypothesis: can we terminate by inhibiting TET? And to test that, if it is true, then we reasoned that if TET2 mutation creates synthetic lethality to further TET inhibition, then IDH1 neomorphic mutations should not coexist with TET2. And we did analyze a lot of data. We showed in a preclinical model that if we introduce IDH1 neomorphic mutation in TET2 wild type, the cell terminates. And then we designed a small molecule antagonist of alpha-ketoglutarate, and it is highly specific to TET. We screened around 19 different dioxinases. It was very specific to TET, more than 10-fold more specificity. And then we found that this is in vitro as well as in vivo. It terminates TET2 mutant clone. And this, while we are trying to develop this as a therapeutic to push it in the clinic, we have been screening a drug library of bioactive molecule, and we found that the L-Thrombopag is a bona fide potent TET inhibitor, and it can work like our specific TET. Apart from activating thrombopoietin receptor, it inhibits TET2 and phenocopies them. And this activation is independent of thrombopoietin receptor activation, because we re-engineered mouse cells putting human TPO, and we saw that and it is very specific to L-Thrombopag, because other small molecule agonist, Evothrombopag or Heterothrombopag does not. So, or the C-Maple agonist TPO itself does not inhibit TET activity in any of the model system we looked into. And we saw that if you look at the TET2 knockout system, L-Thrombopag doesn't work in the mouse or in the human cells, in primary cells, suggesting that L-Thrombopag is a bona fide TET inhibitor which amplifies hematopoietic stem and progenitor cells. And for the more detail, we published this paper a couple of years back in JCI. Thus, I'm sorry, this is completely messed up. This work which I presented today was actually Yarik's has been pushing us for a long time, and I'm the one person who's presenting, but a lot of people in the group who worked on it. There are a lot of people in my lab and then Yarik's lab. And then there has been several reports. We tried to publish. We published this paper as a cover page article in Blood Cancer Discovery, and there's a lot of news around that. The TET inhibition can be a therapeutic modality in TET2 mutant associated neoplasia. Thank you.
Perfect. So thank you very much. You could take a seat on the panel. So just in the interest of time, we'll move on and have questions again at the end of the session. So next, it's my pleasure to introduce a close collaborator, Stephen Chung, who will be talking about molecular relapse of MDS after transplant.
All right. Well, thank you for the organizers for giving me the privilege of presenting at this symposium. I'm going to be talking about work my lab has been doing over the past couple of years, studying stem cells to try and predict relapse after transplant. This is a relatively simple assay, but we think it's quite powerful, and see what you guys think. All right, so work by many investigators over the past 2 decades has really helped to establish that the hematopoietic stem cell is the cell of origin for MDS. And so I won't belabor this point, but really draw your attention to this observation that these stem cells are selectively resistant to all standard non-transplant therapies. So things like azacitidine, lenalidomide, as shown on the right here, when they work well, can get rid of all the disease, molecular disease we can see in a bulk bone marrow. But if you sort stem cells at this time, you can always see resistant stem cells that are persistent. And we think this is really what drives relapse. And so as a result, the only way you can cure an MDS patient is by allogeneic transplant. And it's long been presumed that this works by replacing or suppressing the MDS stem cells. However, relapse after transplant is quite common. It occurs in up to 40% of patients with high-risk disease, and these are, in fact, the patients that we do transplant. There are molecular subtypes like P53 mutated, where the relapse rate is unfortunately much higher. So we asked the question of whether relapse after transplant is driven by MDS HSCs and whether their persistence would be predictive for relapse. And conversely, when patients who are actually cured, are we actually getting rid of these stem cells? So the challenge to studying this, really over the past 10 years that we've been asking this question, has been the limited number of stem cells in biospecimens. So in these post-transplant biospecimens, oftentimes we only can get 100 to 1,000 stem cells. More than half of them actually have fewer than 100, at least in our bank. And so how do you sequence for MDS-associated mutations in these stem cells? In terms of bulk targeted sequencing approaches, if you look at commercially available pipelines, they all require about 10 nanograms of DNA, which is oftentimes 10 times more than what we actually have. And even when these work, you get all this amplification bias and allelic dropout, so you don't really know what you're looking at. And there are additional techniques like digital droplet PCR that, in theory, have no lower limit, but these require a lot of customization for every patient and every single mutation. When you look at single-cell approaches, broadly applicable high-throughput approaches like Mission Bio, give you single-cell resolution, but they are not single-cell input. Even the newest version requires at least 20,000 cells. There are low-throughput single-cell techniques, but these often require a lot of patient-specific customization and can be quite labor-intensive. So these are things that are never really going to scale to the clinic easily. So to overcome this, we didn't invent any new technology, but simply adopted technologies previously designed to look at cell-free DNA and FFPE samples. And the key feature of these workflows is they incorporate unique dual-index primers, or UMIs. In principle, they're pretty much the same DNA barcodes. And we merged these with a hybridization capture panel encompassing 260 myeloid genes. And we also included all HLA loci so we could estimate the donor versus host composition of different cell populations. On the right is our bioinformatic pipeline and a really talented MSTP student who did all this work, Ben Kroger. And our assay, we would note, really scales down to about $150 per sorted cell population, so it's quite cost effective. So when you do low input sequencing, there's a lot of PCR involved. And so bioinformatically, you need to remove PCR duplicates. And this is highly imperfect. It relies on estimation based on the start and stop sites of these DNA fragments. And so you oftentimes lose library complexity from over-deduplication. So if you simply barcode each fragment of DNA before any PCR, as shown on the right in red and purple, then this significantly refines the accuracy of this deduplication. So this is what things look like for the first 200 sorted cell populations we applied this to. And this is with 8 million reads per sample. And what's plotted is cell input by effective UDI-adjusted read depth. So as you expect, if you put in fewer cells, you get a lower read depth. And so we think this is actually a pretty good validation of the accuracy of our deduplication. Now, at the lower limit of this technique, which is about 10 to 15 cells, you get a lot of duplicates. But as shown on the right, at this lower limit, we're actually capturing more than 50% of all sorted genome equivalents. So as an example, if we sort 20 cells, diploid genome, you have 40 genome equivalents, we can get a read depth of more than 20 consistently from 20 sorted cells. So we think the efficiency of conversion of sorted cells to sequenced library is quite high, so we went on and applied this to post-transplant bone marrows. This is simply showing how we sort HSCs from human samples. We initially didn't know how sensitive our assay would be, so we combined HSCs and MPPs. We now separate them out. We always set aside LMPPs because in many patients these may represent an incipient leukemia stem cell, even MPPs in some samples. So these are the first two patients we looked at. They both had biallelic p53 mutations, both relapsed. One relapsed, or we had bone marrows from 52 days and 166 days, respectively, before relapse. And if you look at the bulk unfractionated bone marrow with high-depth sequencing, 400x coverage with the same pipeline, you can't see a single mutated read. But if you sort HSCs and MPPs, as shown on the left, these are heterozygous mutations, so nearly all of these cells are actually involved with the mutations. In the sample on the left, most of the progenitors are involved as well. If you look on the right, this is a good example of a case where you can only see the mutations if you sort HSCs and MPPs. And so we think this really demonstrates that sequencing of HSCs and MPPs is really necessary to maximize the sensitivity of post-transplant disease assessment, and we think this is also really important data suggesting that relapse after transplant is likely driven by HSCs. Now, it is true that if you sequence deep enough, you're likely to be able to detect these mutations, but we would argue that this would require, if you do the math, read depths of 100x or 1,000,000x coverage that would be really financially impractical. And in many cases, you may actually be limited by your library complexity. You may not be lysing enough cells to give you a complex enough library. And we also, as I'll show, have found that that the mutations are in matter. So to validate this assay, we put together a multi-institutional cohort. Dr. Mashevsky, Dr. Solomon have generously provided samples for this, as well as collaborators at Sloan Kettering. So we took patients transplanted for MDS or secondary AML, those with at least a year and a half of relapse, and a bone marrow from within 120 days after transplant. And to be conservative, we only included samples where we could sort at least 45 HSCs or MPPs and get a read depth of more than 20. So our initial cohort that I'll show, it was 33 patients, half of them relapsed. We're now up to about 50 patients and are working on the paper. But this is what we found. So if you can find these mutations in HSCs and MPPs on the blue line, it is 100% specific for relapse. Conversely, if you don't see the mutation in red, it's not 100% sensitive. We didn't really expect it would be from such small numbers of HSCs, but it's about 82% so far. Conversely, if you look at bulk bone marrow, you fully miss in red 50% of patients who will relapse. And additionally, if you look at the blue line, not everyone where we detect a mutation eventually relapses. That curve does not go to 100%. And we think it's because those mutations are not in HSCs, and we actually know they're not because they're the same patients we sequenced on the left. And so we think this really demonstrates that the mutations have to be in HSCs to actually drive relapse. So importantly, the average time to relapse is about 6 months from detection. And we found that in the couple cases that we missed, or if we only detect mutations in HSCs, you have a much longer lead time to relapse than if you can already detect things in progenitors or in bulk bone marrow. And so this really raises the possibility for preemptive therapies such as azacitidine, mutation-targeted therapies. But I think most MDS clinicians here would agree that post-transplant use of these therapies probably has limited effect. Maybe it forestalls relapse. If someone's going to relapse, it's probably not going to prevent it. But I would argue that if our assay is really 100% specific for relapse, which we need to validate, it really gives you clinical equipoise to do some more intensive things, even up to a second transplant. And there's a lot of emerging data that Ven-Aza post-transplant can be well tolerated, but anecdotally, we actually see some pretty good responses in terms of regaining donor chimerism. So that's ongoing work. Let's see if... Okay, so to summarize, our low-input sequencing assay is cheap. It's broadly applicable, doesn't require any patient customization. We actually do de novo mutation discovery with this platform. It's scalable. Our students got it up and running so he can do hundreds of samples a week. And we found using this that persistence of MDS stem cells is 100% specific for relapse. Importantly, persistence of mutations in non-HSCs is not entirely specific, and so with bulk sequencing, you can sequence as deep as you want. It still will not get you that cell type specific information. And for future clinical directions, we obviously want to validate this in larger clinical cohorts, and we're eager to work with anyone who would like us to look at their samples. We'd like to look at the effective transplant strategies on MDS HSC depletion. And perhaps most importantly, we think this enables the performance of really HSC biomarker-driven prospective clinical trials of preemptive therapies with things like second transplant and Ven-Aza. So I'd like to finish up by talking about a little bit of the mechanistic work we're doing. So we're trying to understand if MDS stem cells evade immunologic surveillance by downregulating MHC2, as has been described in relapse after transplant for AML. Recent work by Simone Haas's group has shown that HSCs do express functionally relevant MHC class II. We've looked at this by flow. It's in our sequencing panel to look for genetic loss. And long story short, we really see no evidence of this. So MDS biologically may really relapse in a very different way from AML. So an alternative hypothesis is that MDS HSC persistence could be stochastic. And we think, with myeloablative conditioning, or for people who work with mouse, lethal dose radiation never really gets rid of host HSCs. They're just so resistant to everything. So we have this hypothesis that perhaps MDS HSCs, shown in pink here, if they're simply at higher levels before transplant, may be more likely to stochastically persist after conditioning. To test this, we're using donor and host-specific SNVs present in our panel, as well as HLA polymorphisms to look for the presence of recipient HSCs regardless of mutation status. And what's shown on the lower right is an example of a case where we could see an IDH mutation, as shown on the left part of that figure, only in HSCs in the blue bar at a VAF of 15%. And the other bars are 5 homozygous host-specific SNPs in this patient. And all of this SNPs, we find at a VAF of 25 to 30%. And so this is an example of a case where we think almost all the host HSCs that are left over are actually the IDH mutant MDS HSCs. And so it's findings like these that really tell us that MDS HSCs likely have mechanisms that allow them to preferentially persist. And so to better understand this, we're applying, like everyone else, a bunch of multi-omic technologies to this. And so I'll just very briefly review these and show one piece of data to finish up. So we applied the Mission Biotapestry single-cell DNA-seq, needs little introduction to this audience, and we combined with this AB-seq, which allows us to look at cell surface proteins to infer the cell identity of the cells we're sequencing, as Dr. Yoshida reviewed nicely yesterday. And we used mitochondrial DNA mutations, which accumulate up to 100 times faster than nuclear DNA mutations. And these serve as endogenous cell barcodes that allow us to trace clonal cell populations. And so this allows us to tell the difference between host and donor cells. And because of the pervasive expression of the mitochondrial transcriptome, it actually allows us to infer driver mutation status in single-cell RNA-seq data. So I'll just show one case to finish up that we looked at with all of these technologies. And so this was a case from Dr. Salman of a P53 mutated MDS patient that was transplanted. This bone marrow was 1 month after transplant, and he relapsed 1 month after this sample. And we enriched CD34 positive cells, did single-cell DNA sequencing with our myeloid and mitochondrial DNA panel. And what's shown on the left is every column is a mutation, every row is a single cell. And so the blocks of red on the left and the right are basically the mitochondrial mutations that allow us to tell host from donor. The column in the middle is the P53 mutation. Gray is wild type, pink is heterozygous. And so you can see the donor cells, fortunately, are P53 wild type, as you'd expect. The host cells are mostly P53 mutant, but there's this sliver of gray on the top, so there are some persistent P53 wild type host cells consistent with our stochastic persistence model. So now that we have these mitochondrial mutations that can identify these populations, we're following up these studies with single-cell RNA-seq that will be mutation and host and donor aware. But these are not purified stem cells, but CD34 enriched cells. So we used AB-seq to be able to subset HSCs and MPPs. And an example of this is shown here, where the FACS plots are on the left, and this is what we put into the sequencing on the left. And what's on the right is AB-seq, using AB-seq to do in silico reconstruction of what we can see by FACS, but then now we can know the mutation and host and donor status of every single cell. So zooming in, this is what this looks like. And so in human, 38 low cells are enriched for HSCs and MPPs. My student really wanted to benchmark this, so he purified normal HSCs, spiked them in, stained them, sorted them with everything else, and used SNPs to deconvolute things. So now we validate our in silico gate, if you will. And all of these are color-coded. So if you look at what they are, donor is in red. And what's quite remarkable, we think, is this is 1 month after transplant. All the donor cells are progenitors. There is not a single detectable donor HSC. All the HSCs are host cells. Most of them are p53 mutant, and a subset are wild type. So to summarize, after transplant, wild-type host HSCs can persist alongside p53 mutated HSCs. The p53 mutated HSCs sure seem to preferentially persist, and so using mitochondrial tracing techniques, we're now studying what molecularly makes them different and allows them to do this. But I think what these data really suggest is in some of these early relapses, especially for p53 mutated disease, relapse may not be MDS growing back out, but it may actually be a failure of donor HSCs to actually engraft at all. You may fail to condition the p53 mutated HSCs out of their niche. And so on the upper right, I have this same ABSEQ plot shown. And remember, I said that this patient relapsed 3 to 4 weeks after this sample. We think when this patient relapsed, it was actually these donor progenitor cells just exhausting. There's no donor HSCs to replenish them. So after 3 to 4 weeks, they're not self-renewing. When they exhaust, you lose donor chimerism. The patient's counts drop, and we call it clinical relapse. But the MDS HSCs really never went anywhere. So with that, I apologize for going a minute over on time. I'd like to thank the members of my lab who did all this work. And,
okay, we move on to the next talk. Valeria will tell us about what's new in DDX41 world. As you know, DDX41 germline variants are the most common congenital predisposition factors in adult leukemias in myelodysplastic syndrome.
So good morning, everyone. Thank you very much for having me here, and I'm most grateful to present to this audience today. So my talk was a little bit general, so I tried to put together something that we know, the unknown, and what we are doing in the lab in order to attempt to answer some of the open questions in DDX41. So germline DDX41 mutation are the most common hereditary mutation in myelodysplastic syndrome and acute myeloid leukemia. As it shows in the left, the most canonical configuration is the presence of a germline mutation, a frameshift mutation, the most common D140 frameshift, which predisposes to a second somatic mutation, preferentially R525H, among other missense mutation. But as we can see in this pedigree in the left, that we reported almost 10 years back, like one of the father of this family carried the canonical constellation, but this constellation was only segregated to one of the son, while the other 2 children, they only carry the monoallelic germline mutation. So on the right, instead, is show one of the most comprehensive study from our Japanese collaborator, led by Dr. Hideki Makishima. He sequenced 396 patients with AML and MDS and found a frequency of 5% in MDS, which, as we can see in the bottom part of this plot, R525H was one of the most common mutation. Was one of the most common somatic mutation. But as we can also see from our experience in the lab and also in the clinic, there are a number of patients that they also carry only germline DDX41 or only somatic DDX41. And these cases are unexplained. And here is show a massive genetic analysis that was done recently on 450 individual participants of the UK Biobank, in which the prevalence of non-synonymous DDX41 was estimated as 1 in 129 individuals, while the prevalence of pathogenic DDX41 was estimated in 1 in about 430. The authors also describe that truncated mutation, they increase the risk of developing MDS. And this increase is much higher when a second somatic mutation is present. However, there was no increase in the association with clonal hematopoiesis. But we also know that DDX41 has a versatility of function, is localized in both nucleus and cytoplasm, and in both compartments the function is different. Is in the nucleus that we reported here back that DDX41 co-precipitate with the splicing factor and regulate pre-mRNA splicing, while it's also in the nucleus in which DDX41 is able to affect and modulate the SNO RNA processing, and lots of DDX41 increase the accumulation of R loop, which they seems to be activating the CGAS STING pathway via activation of interferon. So all these mechanisms, they take place in MDS, but why and how each of them is then able to lead to leukemogenesis is not known. So we know that looking at patients and their genome, we can learn about lessons from the nature. And we have also learned that in the case of DDX41, this gene is present on chromosome 5q, but is never deleted, as we know that somatic mutations are often missense mutations. There is never a somatic mutation that might be frame shift or truncated. What we don't know, so this is part of that we are working recently in the lab in trying to understand how to explain the cases that they only carry monoallelic germline DDX41 or the one that they only carry somatic DDX41. And there are other open questions, for example, looking at the structure and the core function of DDX41, can this mutation be hypomorphic? And what about gender? Because we know that there is a prevalence that is skewing in the male. However, we have kind of demonstrated that none of these male patients that they carry DDX41, they have a skewing of mutation on genes of the chromosome X. So what we have done in the lab is to run whole exome sequencing and deep targeted sequencing on over 1,800 individuals with hematologic disorders, and 300 patients, they had simultaneously whole exome and deep targeted sequencing done. And what we were interested on is understanding out of these 154 patients that they carry variable variant in DDX41, what was the first hit in cases that they lack the second somatic mutation, and what was the second hit in cases with only monoallelic germline. Out of these 154 cases, we found 60 cases, very abundant, that they only had one germline in DDX41. 25% of the cases, they had the canonical configuration, so there was nothing to explain. Another 14% of the cases, instead, they carry only a somatic DDX41. Of course, the majority of these patients, they had R525H. So the family that we look more first was actually the family of the early cases, because DDX41 is, of course, the hallmark of MDS AML, but there are other early cases, and these they have been spotted in some pedigree and some family. And we found that a good fraction of patients that they lack a somatic hit actually carry somatic mutation in other RNA early cases. There was a variability of these early cases inside our cohort, and we also found that there were 3 cases with germline RNA early cases when a somatic mutation in DDX41 was present. We also know that part of these cases might have been not analyzed properly if bulk sequencing was used for performing whole genome or whole exome sequencing. In 11 cases that we spotted as candidate, and we thought that these cases, they had the somatic R525H. Instead, we were able to do not detect this mutation. So in these cases, when we manually expected the 3 of these cases, 2 of them, they had the germline D140, we really found a very low reads the presence of a somatic R525H. On the other hand, we also know that several of these cases, they might not, they might have cryptic R525, but the population that is used for whole exome sequencing is actually a bulk population. And indeed, we have done a collaboration with Dr. Steve Chung, and we have demonstrated that DDX41 somatic mutations are present in hematopoietic stem cells and in progenitors and fade away in the differentiation tree. So we also spotted about 40 cases with the other RNA helicases mutation that they did not have DDX41. Male predominance was still a general phenomenon. Variability of this mutation was present also across the mRNA expression level. And some of these helicases are also present in chromosome very important in MDS AML, like chromosome 5q as DDX41 and chromosome 7q. So now how we are trying to investigate a very fascinating process, which is the somatic gene rescue, which we know that is a selection process of a germline lesion, either a copy number or a germline mutation. And maybe DDX41 could be a target of a somatic gene rescue, because this process can actually happen either way when there is an hypomorphic mutation in genes that are very vital for the cells, for the hematopoietic system, or also a gain-of-function mutation in genes that inhibit the hematopoiesis. And in this context, what can happen is that this process can clonally expand the cells in a way to relieve the depressed hematopoietic stem cell pool. And this can span from a discrete, simple hematopoietic stem cell depression up to a frank leukemia. So, but how did we address this, knowing how difficult it is? Also the variability of DDX41 mutation, we decided to use the in silico prediction model when we use R525H as a standard, knowing that was a sweet spot and frequently is mutated in this patient. And we model also other hotspot mutations that they were distal and proximal to DDX41, finding that many of these mutations, according to the location in which they were present in the 3D structure, they're actually able to disrupt the activity of the helicase in the unwinding process, basically working on this small mechanism that they are important for an exchange of ADP and ATP. And this actually was done also for some of the presumed germline mutation. What we did, we predicted in the same way in collaboration with Dr. Chao Yang from the University of Tennessee. And what we were able to see is that there was a cluster of mutation that they bind in a region which is the blue light on the right. And this cluster was very important because it defined the activity of the helicases in collaboration with other partners. And as we can see, there is also a very common non-synonymous mutation, M155 high, that was also yesterday brought up in one of the talks from Dr. Gutierrez. In conclusion, what we know is that DDX41 constellation is the most common and ones that can be most likely explained in patients that come to the clinic, but that there are a number of cases in which the genetic lesion is actually unknown, and we think that in these cases there is a phenomenon of compound heterozygosity with other RNA helicases, but we do not exclude that also other genes can be involved in a similar matter. We also think that sometimes it's important also to apply a more sensitive NGS ultra low input a type of technique in order to detect some presumed case that are actually cryptic biallelic. And of course, somatic gene rescue is a fascinating hypothesis that we are investigating, and we will be all happy to receive feedback for all of you. And this is my last slide, and I want to thank everyone, you for attending, and all my lab, and of course Jarek and all the people that they work with me every day. Thank you so much.
Now is the time for the last presentation of this session. Just recently I attended a meeting and I heard a terrific talk, and I just wanted to top this particular session with a presentation by Satish Nandakumar from Albert Einstein. Thank you and welcome, and thank you for coming.
Yeah. Thanks, Jarek, for the kind introduction. How does it move? Right. Yep. Good morning, everyone. My name is Satish Nandakumar. I recently started my lab in Albert Einstein College of Medicine, and today I'd like to talk about some of our work that we are trying to do on dissecting genetic predisposition to clonal hematopoiesis using genome-wide association studies. So what is clonal hematopoiesis? Clonal hematopoiesis, as many of you know, it's an age-related precursor pre-malignant state that is characterized by expansion of blood cell clones that carry somatic mutation in Mds driver genes. And many of these mutations are thought to arise in the hematopoietic stem cell compartment. And why do we care about clonal hematopoiesis? Clonal hematopoiesis is important because the carriers who have these CHIP mutations have an tenfold increased risk of developing hematological malignancies later in their life. In addition to increasing the risk of hematological malignancies, CHIP also increases the risk of non-hematologic conditions such as cardiovascular diseases, and diabetes, and also it's recently been implicated in neurological conditions as well. So what we know about CHIP so far is these are the common mutations that are found in clonal hematopoiesis. Most of the common genes are in epigenetic factors such as DNMT3A, TET2, ASXL1. Many of these we heard about in today's talk as well. Other mutations are found in splicing factors such as SF3B1, SRSF2, and we also have mutations in DNA damage response genes such as P53 and PP1MD1. Many of these somatic driver mutations have been very well studied and characterized using both mouse models as well as human iPSC systems as well as human CD34 systems, and we have an understanding that these somatic mutations either increase the self-renewal of hematopoietic stem cells or gives a fitness advantage to these stem cells in the context of chemotherapy or other therapeutic conditions. But we think the somatic mutations is not the entire story. Our lab is trying to understand whether germline, there is any germline components to this. Does germline genetic variants also influence clonal hematopoiesis? And how can we identify these germline variants? So our approach is to identify these germline variants by using human genetic studies called genome-wide association studies or GWAS studies. To just give a brief introduction on GWAS, GWAS is a human genetic study. It takes advantage of the common genetic variation in the human population, and this study can pinpoint locations in the genome that are associated with any particular phenotype or a disease of interest. So when you perform a GWAS study, you get these Manhattan plots, and these Manhattan plots contain genetic risk loci, and if you probe deep into these risk loci, you can identify single nucleotide polymorphisms, SNPs, and genes that are play a critical role in the development of this disease process or the trait that you're studying. Are there GWAS studies done on clonal hematopoiesis? Yes, we were involved in the first, one of the first GWAS studies done on clonal hematopoiesis. It was done in 97,000 individuals, and this study identified 3 germline genetic risk loci that are associated with CHIP. Soon after that, 2 additional GWAS studies has been done. On these studies were identified. Right now, we have about 24 genetic loci that has been identified. These with the largest study containing 600,000 individuals. So let's look at what these genetic loci looks like. If you look at in these GWAS studies, the genetic loci, most of these genetic variants found in these studies are in non-coding regions of the genome. The coding variants are extremely low, only 4%. Majority of them are located in intronic regions and intergenic regions. And that makes it challenging to study what are the mechanisms by which these genetic variants act upon. The hypothesis we have in our lab is that because these variants are non-coding, we hypothesize that these germline variants affect gene regulatory elements, and they affect elements in hematopoietic stem cell compartment, or HSCs. The other hypothesis we have is these germline genetic variants also cooperate, likely cooperate with the somatic chip driver mutations such as TET2, DNMT3A, SF3B1, to allow the progression of these mutant clones to expand. So we have some evidence to prove this in our previous study. So I'm going to quickly go over our previous data. So we previously identified a genetic risk variant that is associated with this myeloid malignancy called myeloproliferative neoplasms, or MPNs. This genetic variant was located 12 kb downstream of this transcription factor called GFI1B. And this variant is actually located in the open chromatin region in multiple hematopoietic cell types such as HSCs, MPPs, and mature cells. And we were able to show experimentally that this location is actually contains an enhancer that regulates this gene called GFI1B. So experimentally, what we did was we took 2 CRISPR guide RNAs using similar approaches that Sergei Doulatov, our earlier speaker, discussed before. We took CD34 primary hematopoietic stem and progenitor cells, and we deleted this enhancer using 2 CRISPR guides and deleted this region. And upon deletion of this region, we were able to show that the GFI1B gene expression was decreased. And in addition, we were able to show that when we cultured these hematopoietic stem cells in stem cell maintenance condition, there was expansion of the hematopoietic stem cells based on surface markers. So in addition to the expansion of hematopoietic cells based on surface markers, we also did functional studies by doing colony assays. So when we did colony assays, we were able to show that these enhancer-related cells has increased secondary colony replating ability, which is indicative of increased self-renewal potential. In addition, these cells, when we transplanted them into immunodeficient mice model, the KIT mutant NBS GW mice, they also showed an expansion of the engrafted cells over a period of 18 weeks if we follow them by using edited alleles by PCR of the blood compartment. And where we are taking this study now is we have shown that this variant expands hematopoietic stem cells. Now we are asking the question, does it actually affect myeloproliferative neoplasm development in vivo? So for this, right now, our lab is developing a mouse model in which we have found a very highly conserved region between human and mouse, and we deleted this GFI1B enhancer in mouse system as well. And we found in the mouse blood compartment there is a decrease in GFI1B expression upon deletion of this enhancer. And we have now crossed this enhancer deletion mice to the classic MPN somatic mutation driver containing mouse models such as the JAK2 V6-1F containing mouse model. And we are able to see promising results that these compound mutant mice, which has both the germline deletion and the somatic driver mutation, seems to progress more faster and die much earlier compared to that of the somatic mutation containing mice alone. So this data suggests that there is a cooperativity between the germline variant and also the somatic mutation that happens in this disease. So in addition to this story, we also have looked at germline variants in clonal hematopoiesis itself. So we identified from another GWAS study on CHIP that there is a variant upstream of TET2 gene. This is a non-coding variant 100 kilobase per upstream of TET2, and also located in an open chromatin region, likely an enhancer element. And when we deleted this enhancer region using the CRISPR approach, we were able to see that there is a decrease in TET2 expression, and the loss of TET2 also resulted in increased self-renewal of hematopoietic stem and progenitor cells. So far I've given two examples in which we started from a non-coding variant, and we were able to show that these germline variants affect hematopoietic stem cell function. But if you look, remember my previous slide on the GWAS, there are a lot of genetic loci that has been identified, is constantly being identified by these GWAS studies. And we can't do these CRISPR deletion approaches and functional validation one by one to test all these enormous number of GWAS variants that are being identified. So we asked, what we are currently developing in our lab is, can we use high throughput approaches to functionally identify these relevant non-coding mutations in using high throughput approaches? So one approach we are to study enhancer elements in addition to CRISPR deletion is by doing reporter assays. This has been well studied in the field. So the way the reporter assay works is you have a minimal promoter that drives the reporter gene, and if you clone an enhancer element upstream of this reporter, you get higher reporter expression. On the other hand, if you have a genetic variant in this enhancer, which inactivates this enhancer function, it decreases the reporter activity. And thus, you can test if a genetic variant is inactivating the enhancer. But what we are trying to do is we are trying to adapt this approach into a high-throughput system in which we can test multiple enhancers all in a single experiment. So this assay is called the massively parallel reporter assay. So in this assay, we have a very similar reporter construct with the reporter gene, minimum promoter, and also an enhancer upstream. The only difference we have here is we have an additional barcode region, 15 base pair barcode region, which is located in the 3' UTR of this reporter gene. So having this barcode, unique 15 base pair barcode, which tags the enhancer, gives us the advantage. Now we can multiplex the whole system. So we can clone in multiple enhancers together with their own unique DNA barcodes tagging them, and we can make these pooled library of reporter constructs, and we can deliver these reporter constructs into target cells of interest. And once we deliver these reporters into target cells, then we can look at the RNA and DNA expression in these cells, and by just counting the barcodes in the 5' UTR of this gene. So the way it works is if an enhancer is highly active, the barcode of that enhancer, you'll see more counts of barcodes in the mRNA level compared to that of the DNA level. On the other hand, if an enhancer is inactive, you will see less transcription from this enhancer, and you will see less RNA barcodes compared to the DNA barcode. So by using a RNA to DNA barcode count ratio, you can assess the reporter activity of this enhancer. So how we are doing this to the GWAS? So we have now the GWAS study, the latest GWAS, which has 24 genetic loci. And this GWAS has identified, if you look at the linkage disequilibrium, we have 3,800 variants in healthy in all of these 24 genetic loci. And we are asking the question, which of these variants affect gene regulatory function? So it's challenging to do even the MPRA on 3,800 constructs. So what we are currently doing is we are shortlisting this 3,800 constructs to find out which are the ones that are most likely, test which are the ones that are very highly promising candidates. So the way we do this is we take our 3,500 constructs, overlap with the open chromatin region, and only select the ones which are located in ATAC peaks. So that reduces the test variants that we are testing. So from 3,800 variants, we have about 590 variants which are located in open chromatin region that we are planning to test right now. And we will also include negative control constructs, which are highly associated variants in non-coding region, but are not located in open chromatin region, likely not enhancer elements. So those will be the negative control constructs. So we now have a library, generated a library of 1,400 constructs that we are testing. So the library generation part is pretty straightforward. You order these constructs as 250 base pair regions. We can test them by oligosynthesis approaches, and we can add both the minimum promoter by PCR method and also the unique 15 base pair barcodes by PCR method, and then we generate this pool library of 70,000 constructs with 50 barcodes each for all these enhancer constructs. So we are currently in the process of delivering this library into our target cells of interest, which is the CD34 positive human hematopoietic stem and progenitor cells, which is the target cells for clonal hematopoiesis. And our plan is to first deliver this lentiviral library, and after 3 to 4 days of infection, we will extract the DNA and measure the barcode counts in these cells. So what type of data we can expect from these studies? This is one of the MPRAs we did several years ago in the context of red blood cells. So we have a genetic variant located in non-coding region. We find that upstream of this gene RBM38. So we find that if you deliver this constructs into K562 cells, both the T allele and C allele show similar activity. But if you differentiate the cells into red cells by adding GATA1 overexpression, we see that there is a C allele has much more higher activity than that of the T allele. So by using this identifying these differentially expressed constructs, we can identify the variants. So these are two additional variants that we tested before. Does this assay works in the hematopoietic stem cells? Because this previous work was done on red cells. Yes, it does work. We have tested a few stem cell specific enhancer elements such as the 3Q21 GATA2 enhancer and also other enhancers such as PU.1. We can see that these enhancers show high level of activity compared to minimal promoter. And when you put in a mutation, the enhancer activity is decreased. And they work in cell lines as well as they also work in primary CD34 hematopoietic stem and progenitor system. So to summarize the work, so we hypothesized that these germline genetic variants also play a critical role in clonal progression of CHIP. And we are identifying these non-coding variants by using our reporter assay called MPRA. And once we identify these variants, we still have a lot of validation studies to do. We will have to delete the enhancer elements, find out what is the endogenous target genes of this non-coding elements. And we hope that by identifying these risk alleles, we can either stratify the patients who will progress to MDS or also find therapeutic targets for these clones. With this, I'd like to thank members of my lab. Many of the MPN work was done in my previous postdoctoral fellow lab in Vijay Sankaran's lab. And with this, we'd like to take questions. Thank you.
Yep, perfect. So now we have about 10 minutes to have questions for any of the speakers. So I see one in the middle, and I'll just take one in the interim. Sergei, I had a question with the check inhibition. I think sort of splicing is this big group of mutations that we've kind of failed to target clinically, even a couple exciting drugs preclinically. And I guess my question is do you think, and have you tested in, let's say, AML PDX models the check inhibition? Because SF3B1 is rare isolated mutation once you're to more advanced high-risk MDS AML. Do you think it's better served in sort of a CCUS, low-risk MDS? Just thoughts on that.
Yeah, that's a great question. Thanks. We haven't tested it, and I think that's where it would be useful is in a high-risk scenario like in a SF3B1 AML, which are rare but very high risk. So that's definitely the next step for us is to keep going with this. Thank you.
So I have a question for Dr. Jha. I'm here. So thank you for showing your beautiful data. So I'm wondering if you have a TET2 heterozygous mutant model. So you compare the TET2 wild type and homozygous, right? So but a lot of patients show TET2 heterozygous mutation. So I'm wondering if even the TET2 heterozygous mutation shows like affect MSH6 or just shows like similar to wild type.
Oh, thank you. I'm sure I was moving fast. We did in heterozygous 2. In heterozygous, the MSH, the mismatch repair complex is not compromised because of the existence of one allele and the expression of the N-terminus of TET2.
Yeah. I'd like to ask Dr. Chung a question. Do you think the failure of the donor cells in the setting of p53 mutations, what's the mechanism of that? Is it some inhospitable microenvironment situation?
Thank you for the question. I think it's still early days in terms of really digging into the kind of what makes those cells different from, say, the host from the donor cells in the setting of p53. If I were to guess, I would probably go for the most simple explanation, which is just that based on what we see, maybe there's no niche for those donor cells. And p53 mutated MDS HSCs may just be largely unaffected by conditioning.
I think that's certainly what we see clinically.
But you avoid, or the HLA part might also allow you to, are p53 mutant stem cells immunologically privileged to be spared by graft versus leukemia effect? This would be another possibility.
Right, and that's the follow-up question is, there's a few people who don't relapse after with p53 MDS, like in Coleman's work. Is it HLA based, or is it some other, or those p53 clones more sensitive to the conditioning? We don't.
Yeah, in those cases, which we have captured some of them in our bulk sequencing, we actually don't detect the stem cells at that point, and so so we're interested in looking at single cell resolution. We have a couple of those cases kind of cooking to see if there's host stem cells that are persistent, and it's again whether it's all just stochastic, and if pre-transplant, if you were able to reduce the burden of MDS HSCs, is that what matters the most? So we really want to look at those cases, because I showed, I think, the worst possible case, but which unfortunately we have a number of. It seems to be like half of the cases we look at.
It's the good ones we need to study, right?
And the good ones, exactly. So we have at least 2 or 3 of those that we're studying further, where we don't see any mutations, mutated HSCs early post-transplant. By bulk sequencing, we're looking deeper to see if first, is it, if we can find pre-transplant samples, did we just deplete more HSCs before transplant? Or is there something about in those cases, do they have an inability? What we're actually looking for is the cases where there's a little bit left over and they eventually relapse, because then we can actually compare the p53 mutated HSCs with the wild type in the post-transplant setting to see what's actually different about them.
So, Satish, the GWAS for clonal hematopoiesis would also give us predisposition factors for actual leukemia or myelodysplastic syndromes that evolves from it, right? So would the signal be not expected to be magnified if you would do GWAS, which would be, of course, maybe more difficult because of the number of patients, but if you focus on patients who actually develop leukemia?
Yeah, I think that's it.
Here we go.
That's a great question. I definitely agree with that. The genetic variants, some of them will definitely lead to progression to MDS AML. GWAS and AML has been done, but it's very small cohort, and it has probably these GWAS identifies like one loci, HLA, some of them are. And I think instead of that, we can experimentally test them. We can test these genetic variants we find from regular chip GWAS and see if these variants allow leads to ineffective erythropoiesis or stem cell expansion phenotypes.
Got it. Yep. Because at the end, it is possible with treatment-related chip, we have always a problem to conceptualize whether treatment by itself creates more chip or whether it accelerates progression to chip. And I think intellectually you have the same conundrum here, whether these variants are variants that accelerate chip, but like things like ATM could be actually the things that gives you more chip. Okay. But right?
Yes, definitely.
Yeah, go ahead. Oh, go ahead.
Yeah, DNA damage genes are hits from the chip, such as ATM and CHEK2 are germline predisposition factors for clonal hematopoiesis, and we have the same.
Go ahead, Amar.
Yeah, this is a question for Dr. Chung as well. So Coleman Lindsley showed very nice data that 33 out of 35 patients in the BMT-ATCN trial went to transplant with detectable TB53, and still we had 20% survival rate in those patients. So how do you reconcile this with the idea that persistence of these, if this is 100% specific, and you still show 20% of patients who did not have persistence still relapse? So how do you reconcile all of this?
Yeah, I think the main difference maybe, and we would really be very interested in looking at cohorts of patients that have matched pre and post-transplant samples, because again, the clearance in, or the lack of clearance and then reasonable clinical outcome in that paper was all looking at pre-transplant samples, so we don't really know the effect of conditioning on reducing those cells. And we would presume that conditioning probably is preferentially ineffective at reducing p53 mutated HSCs, but we don't actually, that analysis hasn't really been done to see kind of a pre-post transplant, and so perhaps in some patients it's just the conditioning regimen that's more effective. At such an early time point post-transplant, we've kind of presumed that the immunoevasive effects should be or are likely to be minimal, but it doesn't exclude that either, and so we don't have a great answer to that question, but it's something we're thinking about a lot.
I think I saw. Was there one more quick question? Yeah. Just for the microphone, 1 second. Sorry, she's coming down.
My question is to Dr. Chung. Wonderful presentation. Again, exactly to what Dr. Zidan said. From our data at MD Anderson also, we found that the long-term survivors of patients with high burden TP53 MDS were those who were transplanted. And a lot of them, though they attained some form of marrow remission, aplasia, and flow cytometry MRD negative, were still having TP53 through on just bulk sequencing. So 2 questions. One, how could we use this approach to understand patients with MDS and maybe even AML subsequently who could be selected and have the highest chance of post-transplant durable responses? And 2, have you seen patients post-transplant who had the TP53 in the hematopoietic stem cell clone where the burden progressively reduced with time as immunosuppression was tapered, etc.?
Yeah, great question. And I think, again, for selecting patients pre-transplant, there's, again, this recent report that clearance of p53 may not be that important. Prior work from Dr. Solomon suggesting that it may be important. So I don't get the sense that that's like a clearly solved question yet, but one of the things that our assay may add some really context to is what cells those p53 mutations are in, and so if you look at p53 clearance or reduction of HSC burden pre-transplant, is that going to be more allow you to identify those patients who will benefit from transplant? We don't know. We'd love to look if people have cohorts of patients. We have just a small cohort at our own institution, so I think it'll really take collaborative efforts to answer that question. But in terms of, so your second question, clearance of, so in patients with persistent p53, do they ever clear it spontaneously? The answer is no, we've never actually seen that. So if we see it a month after, they always eventually relapse. If we only see it in the HSCs, I didn't have time to show this, in some cases it does take like a year and a half, 2 years to actually relapse, so there is some lead time. What we do see is that in patients with p53 who get some form of clinical relapse, they lose chimerism, drop their counts, give some reason for the clinician to intervene. We've seen in at least 2-3 cases, Ven-Aza actually allow for restoration of donor chimerism. So we think, we don't know exactly what it's doing, and we're looking at this at the HSC level right now. We've a number of samples. We'll get back next week to have the answer to that, hopefully, but spontaneously we don't see it happen.
Thank you. Just another part, maybe we can discuss this later, but have you ever seen donor-derived TP53 in some of the primitive cells and that expand with time and maybe drive relapse?
That's a great question. We haven't seen that, actually. Yeah, we haven't seen any donor-derived chip, actually, which we expect if we keep on looking, we should see it.
And just a brief comment, I mean, this actually may be a place where early HMA maintenance, again, where you have this persistent small LSC, we're actually working on some deep sequencing data in a post-transplant maintenance, and we have had patients who you have this very low burden, and you can actually serially see it decrease. So although, again, obviously we'd like more targeted approaches, but the HMA maintenance in this exact setting may actually be quite beneficial, especially very, very early on in the post-transplant setting. I think unraveling those mechanisms needs to be done as well. So I think with that, I think a really great morning session. So it looks like we have about a 10-minute break till we start up. And so thanks for all the discussion.